Welcome to this overview of steroid hormones, where we'll identify their nature, categories, synthesis sites, and mechanism of action. Steroid hormones are a distinct class of chemical messengers that play vital roles throughout the body. These messengers are characterized by their unique chemical structure and their lipid-soluble nature, setting them apart from other hormones. Let's look at the foundational properties that define all steroid hormones. All steroid hormones are lipids, meaning they are synthesized directly from cholesterol, which is their common precursor. They are lipophilic, or lipid-soluble, and consequently, they are poorly soluble in water. Because they cannot dissolve directly in the watery blood plasma, steroid hormones circulate throughout the body, bound to specific carrier proteins. Steroids are divided into several functional categories, each with distinct roles. Glucocorticoids are primary examples, centrally involved in regulating glucose metabolism and managing the body's stress response. Mineralocorticoids manage the delicate balance of electrolytes and water within the body, primarily in the kidneys. The sex hormones, produced mainly by the gonads, are essential for reproductive functions and the development of secondary sexual characteristics. Even vitamin D, specifically vitamin D3 and its active metabolites, is classified as a steroid hormone. Steroid hormones are only produced in a few specialized glands throughout the body. The adrenal cortex is responsible for synthesizing and releasing both glucocorticoids and mineralocorticoids. The gonads, which include the testes and ovaries, are the primary sites for the production of the sex hormones. The placenta is another critical site of synthesis, producing various steroid hormones necessary to maintain pregnancy. Unlike water-soluble hormones, steroids act via a unique mechanism inside the cell. Being lipid-soluble, they easily cross the plasma membrane to bind to receptors located inside the cell, either in the cytosol or the nucleus. Once bound to their receptors, they typically trigger gene activity, stimulating the transcription of specific proteins that carry out the hormonal effects. Due to this genomic mechanism, which involves changing gene expression, the overall response is generally slower than that of non-steroid hormones.